So it's a great pleasure to be here, not because it's just Trieste is such a beautiful place, and it's a great pleasure for me to introduce our studies, which we did last several years, and to speak in the front of you and hope that you, at some moment, you are going to use and remember this kind of presentations. So, in comparison with Gino Morris, actually, I want to say thank you to you too, because you made very good introduction to the field, because this is structural biology field and it's very heavy stuff. And in comparison, there are two significant methods like cryo-EM and Xray. Basically, I'm going to speak only about Xray, because we solved the structure of prokaryotic ribosome working in United States together with Harry Noller and Jamie Kate and my wife Marat Yusupov. We were 4 people in this determination first. And then we solved the structure of eukaryotic ribosome working in department of Gino Morris in Strasbourg when we came with my wife there 10 years ago. The important thing for you just to imagine how heavy it is, we spent about 15 years of biochemistry work for preparation and knowledge and everything, collection of knowledge in order to crystallize the ribosome, and then structure determination was during 1 year. Here we spent, as you can see, about 10 years of biochemistry work to collect everything together, and then in 1 year we solved the structure. But what it means solving the structure in 1 year? It means that three people from my group sitting in dark in a computer room during 1 year, building every molecule of this huge complex, and then finally we have Xray structure, which is quite different from cryo-EM stories, because today people, what they do, they solve the structure of ribosome complexes on cryo-EM, and it takes usually about 2-3 months for the structure complete and publication. It is different, but they use our knowledge. They use that structure to put in the structure of cryo-EM. So that's why this is important. This is just to increase the importance of the work what we are doing. So yeah, first I forgot to tell you that I'm going to stop in the correct time, so we will have a lunch in a good time, no problem. It's just a question that I have 2 presentations here, so if I will not finish my talk today, I will continue next time, okay? So I'm going to speak a little bit about the history of question, because the question is very difficult, and the protein biosynthesis problems start the structure of ribosome in protein biosynthesis become interesting during long time, and people trying to use different type of approaches to understand what it means, the ribosome, the major machinery of protein synthesis. And in the beginning of our study, which was 40 years ago or 35 years ago, the information about the ribosome structure already was quite good. This is the model of ribosome, and you can see here that 2 important things, that the ribosomes have 2 ribosomal subunits. One is large, another one small. And first of all, what was recognized here, that both subunits, they are not symmetrical. That principle differences from the ribosome complex as RNA protein complex, different from viruses. When we speak about virus has internal symmetry. Here there is no symmetry. More than that, if to imagine that what kind of images people use for such interpretation, I still have difficulties today to imagine how they went from this to this kind of picture. But they were absolutely right. Today we know that the structure, it is like this. It's just low resolution, very low resolution. Another important thing is, again, development of methods. This is pure biochemical methods, electrophoresis, and And again, 40 years ago, a little more than that, people in Günther Wittmann laboratory, his name is Carl Schmidt, developed two-dimensional gel for separation of every ribosomal protein. And for the first time, they show that for bacterial ribosome, we have 50 individual proteins. Again, in comparison with virus, in virus case, usually we have one code protein. In RNA, for example, RNA case, RNA inside. Oops. So the third thing which is very important for interpretation of ribosome structure was the long story of the sequence analysis of ribosomal RNA. Two ribosomal RNAs from small subunits and from the large ribosomal subunit, they are the biggest RNAs in the cell. Each time, if you take a cell, make extraction of RNA, put on the gel, you have two bands. Okay? This is 23S RNA and 16S RNA in bacterial case. The fight for sequence was long time. One sequence came out from the Strasbourg again, which we come from, and another sequence from Harry Noller laboratory. That was very important point because Harry used new technology for sequencing of RNA, and then they predicted secondary structure of RNA, which is also surprising today, absolutely correct. At that time, they predicted RNA, which we found in the three-dimensional structure of ribosome, and we show that was correct prediction. If we speak about crystallography, the major thing is crystallization of ribosome. And of course, it's very difficult to imagine that non-symmetrical ribosome will be crystallizable. And it is such a big complex RNA, as I can show to you. It's RNA and proteins in bacteria case. The size of the ribosome is about 2.5 million Dalton, which is huge organization. But here we were lucky. Not only us, of course, science in general was lucky. And the reason is that was phenomena found many years ago that crystals in the cell, in eukaryotic cells, in chicken embryos, for example, as I show here, they can form two-dimensional crystals inside of cell in stress conditions. So simply, we take the eggs, put them in a cold room for one night, and next morning we take the embryos and look in microscope, and we will see ribosome crystals. Not three-dimensional crystals, but they ribosomes like that, they survive in stress conditions. So these two guys, Milligan and Unwin, later, they demonstrated that this kind of two-dimensional crystals is possible to isolate and make them three-dimensional. Three-dimensional means we are very close to X-ray crystallography. Sorry, this is not that. This one is correct. So second person which was very important in this field was Ada Jonat. And Ada Jonat was working in Günther Wittmann laboratory, and her laboratory was able to get three-dimensional crystals from large ribosomal subunits of Bacillus thermophilus. And for the first time, they demonstrated that these three-dimensional crystals can give the diffraction. And they show that we can use crystals of ribosome for X-ray structure determination. Later, they improve quality of crystals, and they show that it's possible to get diffraction until 3 angstrom resolution, which means that ribosome will be, at that time, would be interpreted. Is it correct in English? Yeah. Would be interpreted until atomic level. So yes, just coming back. It's vice versa. So this is our contribution to the field. And here I want to say once more thing that, of course, we have kind of parallel development of methods like Xray crystallography, which Dino Morris in his talk demonstrated how it was developed during last 50 years from one type, or more than 50 years, of course, but in my vision 50, okay, because I'm not so yeah, because in the history of, there is a very big history of X-ray. I'm not going to talk about all this, but in my time, it was development of X-ray from very poor synchrotrons with data collection on films to the extreme synchrotrons what we have today with the very powerful beams, and the data collection takes just a minute to collect full data set for the crystals which you are studying. So during this time, in parallel with the synchrotron development, was very important development of biochemistry. As I said, that in our case, when we were doing crystallization and crystallization, it doesn't mean that we just try to do crystallization. We study ribosomes, how they behave and how they put them at conditions of crystallization, and it took really long time. So we introduced thermus thermophilus as a basic material. Thermus is extreme thermophile, which helps us to get first crystals of 30th ribosomal subunit, and that crystals were then used by two laboratories of Venki Ramakrishna and Ada Yonat to solve the structure of small ribosomal subunit, and they obtained Nobel Prize later for this work. So we were not right. We didn't work on 30th subunit. We went to the full ribosome. And with the full ribosome, this type of crystals never diffract very well, so they diffract at 15 angstrom resolution, which was not really interesting. And so finally, as I said, we solved the structure of full ribosome that was already complex, containing tRNAs and messenger RNA in, together with Harry Noller and Jamie Cate. And the final work was, one more work was Saccharomyces cerevisiae ribosomes for the high resolution of eukaryotic ribosome structure. Just coming back to summarize what I said, that this is the ribosome structure, X-ray ribosome structure. Here, maybe it's better I will show you just moving. Yeah. So we are speaking about the full bacterial ribosome, which has two ribosomal subunits, and this ribosomal subunit, basic part of the ribosome, is ribosomal RNA, which presented here in gray. Okay, the colorated pieces, this is individual ribosomal proteins, and inside of the ribosome we have tRNAs and messenger RNA. I will show you just later. Okay, as I said that the core part is the ribosomal RNA and individual proteins. In bacteria case, we have 50 individual proteins, and inside you can see transfer RNAs and messenger RNA. So by dissociation of subunits, you can see this. These ligands, 3 tRNAs, they interact with messenger RNA by codon-anticodon interaction on small ribosomal subunit. First of all, messenger RNA located there, and all these ligands are located in interface area between two ribosomal subunits. So when we speak about ribosome, so now we shift from three-dimensional analysis of the ribosome to schemes, which is common for biology people, but it's easy to see what ribosome is doing. So I'm going to speak about incorporation of one amino acid in a growing peptide. So this is schematically ribosome, two ribosomal subunits, messenger RNA, interface area between two ribosomal subunits, and here we have 3 tRNA binding sites, A site, P site, and E site. In P site, peptidyl tRNA with a growing peptide. The new amino acid comes in a complex with elongation factor TU, and binds to aminoacyl tRNA binding site according to codon-anticodon interaction. Okay? Then after GTP hydrolysis, TU factor leaves and tRNA goes for accommodation. They accommodate to the ribosome where two CCA ends become very close to each other in a 50S ribosomal subunits in peptidyl transferase center. Here we have peptide bond formation. After this process, we have tRNA with peptide growing in A site, and P site tRNA become deacylated. Now all ligands have to move from one side to another, and this process catalyzed by elongation factor G. Then tRNA elongation factor G binds to the ribosome and catalyzes translocation. After GTP hydrolysis, factor G leaves, tRNA deacylated from E site leaves, and we have initial situation like it was before. And this is cycle, which happened many, many times, depending from amount of amino acids in a synthesizing protein. So as I said, that we are structural biology people, so we study ribosome structure as with the functional elements in, and this is simply ribosome with all the important compounds which we have. Why I say that? Because there is a phenomena, ribosome non-enzymatic protein biosynthesis. Non-enzymatic means no factor involvement. Normally, ribosome can produce protein without involvement any factors. Of course, I'm talking about in vitro system. So it means that all mechanisms which we study, we can study without involvement of factors, only by these three compounds: ribosome, tRNAs in 3 sites, and messenger RNA. So now we have a structure of Xray structure of the ribosome, and we have before, 20 years before, we have a structure of tRNA, which was determined in Dino Morris laboratory 20 years earlier. So now we can combine everything together and study ribosome function by formation of different type of complexes and structure determination of them. So 15 years ago, when the structure of first bacterial ribosome have been determined, and this was presented in Cold Spring Harbor ribosome meeting, after that practically all groups who are involved in ribosome structural biology business, they start to analyze complexes of ribosome with inhibitors. Bacterial ribosome inhibitors, we know, this is antibiotics, and that was very interesting to learn how they work and suggest the mechanism of this inhibition of ribosome function. More than that, I want to say that more than about 50% of known antibiotics today, they are the target of these antibiotics is the ribosome. So that's why it is so much important to learn. Here we see elongation cycle, which I just showed to you, binding of tRNA, peptide bond formation, then translocation stage. As you can see that every step of this mechanism divided for small steps have a list of antibiotics. Just by interaction or disturbing this stage, they can switch off the translation of the protein production in the cell, and cell will die. And most part of them are specific for bacteria. That's why we can use it to treat our body, ourselves when when we are sick or sick by bacteria. So just to remind you that there are 3 important functional sites on the ribosome. One is decoding center, which is in A tRNA binding site. This is the recognition of tRNA in decoding center where the tRNA and messenger RNA form codon-anticodon triplet interaction. Second, the important place where peptide bond formation takes place, which is on the large ribosomal subunit. This is peptidyl transferase center. And then from peptidyl transferase center, we have a tunnel for the growing peptide, which will go through from interface area to the solvent site of the ribosome. This is peptide tunnel. First analysis of antibiotics. They show the group of antibiotics which interact with tRNA and messenger RNA binding sites. This is the interface of small ribosomal subunits with messenger RNA. This is magnification of the picture. This is messenger RNA place and the tRNA binding site A and P. You can recognize several antibiotics like tetracycline or streptomycin or iodine or kasugamycin. This is the known antibiotics. Maybe you don't know them because in the past, for I don't know, 40 years ago, the antibiotics, they had original names. Today, pharmaceutical companies, they changed the strategy and they sell the same antibiotics with the trading names. So that's quite difficult to recognize that we use the same type of antibiotic because there are very limited amount of antibiotics exist. And the problem today, the antibiotics which people use, they become, the bacteria become resistant against these antibiotics. And pharmaceutical companies, actually, they use classical antibiotics to change them and make them still efficient against resistance of bacteria. This is another type of group of antibiotics. They interact with this is tRNA. Okay, this is slice of the large ribosomal subunits on the level of tRNA in P site. This is growing peptide through the tunnel, and group of antibiotics interact in peptidyl transferase center and beginning of the tunnel. You can see here also several known antibiotics like puramycin or macrolides and so on. I will show you again one more movie demonstrating how antibiotics inhibit the ribosome function. This is initiation complex of the ribosome with messenger RNA, and as you can see, okay, now with the transparency you will see how it works. So the tRNA non-enzymatic case deliver amino acids and binds to the ribosome, and after peptidyl transferase reaction, the incorporation of amino acid happened according to the codons of messenger RNA here. This part is decoding center, as I explained before, where the tRNA first interact with forming codon-anticodon duplex. If we have in the same place we have tetracycline, tetracycline binds to decoding center, and the tRNA, which comes with a new amino acid, has difficulties to form codon-anticodon interaction. So simultaneously, tetracycline and the new tRNA cannot bind to the same place. So in the presence of tetracycline, we stop the protein biosynthesis. But tetracycline doesn't have the high affinity. Tetracycline can stay, can move. If it moves, then still protein synthesis can continue. On the top of it, as I said, with not only low affinity, but the bacteria develop some kind of resistance against tetracycline. So what pharmaceutical companies are doing, they are taking this kind of initial antibiotics and developing modification of it against this resistance. And for example, today we have third generation of tetracycline, which has named tetracycline with that modification. And we study these interactions of tetracycline and tetracycline on the ribosome and show that the tetracycline has just one additional interaction on the ribosome. And that increases significantly the affinity of tetracycline on the ribosome. And on top of that, tetracycline does not have resistance for the moment. Bacteria, they don't have resistance against tetracycline. That's why the development of new drugs, the important thing, and they based actually at the known or known antibiotics. But I said that one thing is antibiotics, another thing is eukaryotic ribosome. Eukaryotic ribosome inhibitors are also very important. And first I would like to make a short introduction. What's the difference between eukaryotic ribosome and prokaryotic ribosome? So one thing is in secondary structure of ribosomal RNAs, we have additional sequences in red which have named expansion segments of ribosomal RNA. So ribosomal RNA become bigger than in prokaryotes. In terms of proteins, instead of 50 individual proteins, we have 80 individual proteins. And if to compare them with bacteria, we see that about 34 proteins are common, conservative for bacteria and for eukaryotes, but we have specific for bacteria 23 proteins and 45 specific for eukaryotes. This is just to general knowledge about the composition of eukaryotic ribosome. In case of eukaryotic ribosome, the structure which we compare is Saccharomyces cerevisiae, which is normal bread yeast. Here in this picture, you can see in red additional eukaryotic elements which appear on the top of bacteria ribosome. In gray, we can see core part of the ribosome, which is quite conservative in bacteria and in eukaryotic ribosome, and all additional elements, in red here, they appear on the surface of ribosome. If to look at interface area, for example, peptidyltransferase center in gray, so it means that it's very similar in eukaryotes and prokaryotes. In decoding center in gray, very similar in both systems. But how antibiotic is able to distinguish between our ribosomes and the ribosomes in bacteria? So it means that there is another level of sensitivity or differences between two systems, which we cannot say just by analysis of sequences and sometimes sequences, but just general analysis of the ribosome. So we need to go much deeper in, and then we will see differences. On the top of it, if to compare two systems in protein biosynthesis, you can see here protein biosynthesis process in prokaryotes. Here initiation, this is termination and recycling, and this is elongation cycle. And if to compare with eukaryotic system, initiation and termination and recycling, we can see that only elongation is common. But still, as I showed you before, one more figure, how these small molecules are able, in this common process, is able to distinguish between bacteria and eukaryotic ribosomes and attack them. So this is a scientific question for us. And should I stop?
Yeah.
Okay, good. I stop. I will continue next time.
Also,